And we will now start the panel discussion, and we'll try, with the help of each of our speakers, to draw conclusions related to regulatory issues. And I must say that I found some, but I have difficulties to find for every presentation. If I may start with, I speak under your control, Professor Salonen. I think you said very important things in this issue. First of all, you reminded us that claims were not, except for one, as far as I know, claims were not accepted or had no favorable outcome in the EFSA evaluation process because the end points were too generic. Okay, that's one point. The other point is that what you said seems also very important, is that change in the microbiome per se cannot be considered as a health effect. Okay? So that means that we have to assess functions. And this is a difficult issue. What do you think about that?
Yes, indeed. I think the whole community agrees that assessing microbial functions is extremely important to get forward, but we do have some technological challenges ahead. First on generating high-level data, and I think even more on interpreting the data, because it's making a biological story of in a way, there is a phrase that no more than a dictionary is a novel, not equally the sequences or the protein peptides are not biology. So we have to interpret them in order to understand what they mean. And I mean, there are active attempts onwards on this field, but I think it will take a few more years that we will make like big jumps ahead in this area.
Having listened to the contributions we've had this afternoon, which are very different, but in some ways are quite similar, I'm trying to pull out a common thread. And one of the threads I've got is potentially a move towards personalized diet. And what I'm interested in is how one might draw from these exciting areas of science, which is still going forward, how that might be included into the concept of personalized diet, and what that does from a regulatory point of view or a risk assessment point of view. And I'm not sure if you can answer the question, but I'd invite you to try. And maybe since Anna has just answered, can I start the other end with James?
I don't think we're even close to being able to identify, I'm sorry. I don't think we're even close to achieve that goal. Namely, every human being is genetically different to begin with. Secondly, the Eskimos' diet and my diet, based on a Hungarian cuisine, okay, and all the other factors that could influence the microbiota, the drugs I'm taking, the aspirin I'm taking, and so forth and so on, is going to be so complex to have my physician or my nutritionist give me that right collection of microbiota that will be perfect for my total context, my genetics and everything else. That's my answer. it's a noble goal, but I don't think we're even close to that now.
Thank you. Richard, any thoughts?
Well, I think I would agree with that. I think it's a great idea, but I'm trying to think how you would introduce it in any workable way, unless you're talking about specific ingredients that you personalize as opposed to the diet per se. And then I guess we're then into the sort of the supplements type of thinking, which I tend to steer very clear of and think it's mainly for making money, not making good health. But I guess if you can do it and do it in a very designed sort of way, then it's a way forward for the future. But I'm just not sure how easy it's going to be to actually come up with something that you can shape that accurately.
Thank you. Plenary?
Well, I tend to agree with what has been said before. Just to add a note of caution there, I think we talk about diet, sometimes meaning a complex set of nutrients that we take. And experiments in experimental setups are often carried out with one particular one, or a few of them added together, and we completely ignore the additive or whatever effect of combinations of diets, which of course we're not ready to assess yet. We have an experimental model system which I cannot describe very well, is not my own work, but is done in collaboration with another group, where we have a genetic defects which brings flies to have a mucolipidosis, which is a disease that happens rarely in children, with lipid accumulation. Well, if you feed these flies, the larvae, with butter, basically, with medium saturated fatty acids, you rescue the genetic defect. So, of course, you wouldn't want to go around and advise people to eat butter. So then, obviously, the problem is that it's still too early to be able to make complex statements about diets in general. To me, I think what is also most important is how the calories are distributed throughout. As we were saying with Anne before during her talk, it seems to me we, in general, especially in the Western world, we eat too much. So it's just the amount also of food that we take and the way we eat, because we have undertaken this habit of eating continuously, which definitely is not physiological. Beside what we eat, it is how we eat. And I think that is probably something that can be easily studied in a little bit more complex setup, both experimentally and on human populations. Thank you.
Anna?
Yes, I could maybe repeat what was mentioned earlier this morning in the session of new diets or nutrition. The keynote speaker there made a very relevant point that the human gene nutrient interactions, they account typically maximum of 5% of the variation, and we know that when we talk about the effect of gut bacteria, we have some estimates that for example, if we measure how much certain diet explains on the total variance of gut bacteria, it's around 10% only in humans, because we have our life history. I mean, in animal models, it's like 60% or something like that, but not in humans. So my point is that, and repeating what was the speaker in the morning made a point, that probably on the population level, the nutritional guidelines don't have to take that much into account the personalized nutrition, but of course on the individual level, on the well-being and also I think safety in the end for individual, it will help if we can use either genomic, epigenetic, microbial factors to stratify individuals so that the person can, without trying all possible diets, to find a tailored food that suits your system.
Thank you. Thank you. I think we have a question from the Twitter feed, so if I can pass to my colleague over there. Could you read it out for the panel?
Yeah, thank you. It's not a question from
Okay, microphone. Thank you.
It's not the Twitter feed, it's from a webcast, so
Okay.
from our online audience, and it's a question from Hungary, from Anna Pavlovic, from the Hungarian Food Safety Agency, and she's asking a question that may be actually relevant for several, if not all, panel members, and she's asking, can epigenetic changes be reversed?
Maybe I can take this. Well, of course, yes, they can be reversed. It's amazing. I mean, if we go back to the best example of epigenetic changes that, in a way, have been transmitted through generations, are the one during the Second World War when populations which were starved had a typical trait of starvation, and that was actually transmitted to their subsequent generations. And now you start to see that there is a tendency to the reversal because, of course, the diet has changed. So they can be reversed. Of course, it depends if what you inherit or what you transmit, or within a single individual life, what is the change that you have. It may be a change in a profile of DNA methylation or acetylation, or histone methylation or so, that can be reverted. But it can be, for example, leading to a complete reprogramming of the genome with a set of genes which were not, for example, highly expressed to be now highly expressed. And that, of course, may not be reversed so quickly. But in principle, yes, they can be reverted.
Thank you.
Let me just add to what Dr. Terra has just mentioned. In general, all these chemicals that have been classified as epigenetic, they don't cause irreversible damage like mutations, but they can be irreversible in one set of conditions. That's in utero. And the best example is the drug thalidomide. Thalidomide was designed to be a great sedative, and to this day, it's one of the best. However, if you give it to a woman who's pregnant at a critical time in the fetus's development, it blocks angiogenesis when the bones are starting to develop. The arms and legs need blood vessels. And if you have the baby who's born now without arms and legs, you can't reverse that. All right? So under those conditions where epigenetic agents affect differentiation of stem cells, if it occurs at a critical time in development, usually this is the critical period, not in me, but in a baby, this is irreversible.
I should actually add to that in that situation, it's the effect that's irreversible, but if there's an epigenetic change, in theory, it is inherently reversible.
Determine the differentiation.
Well, once again related to regulatory issues, this morning you showed models of animal models to investigate the possible effects of bacteria on, I don't know, gut and other organs. And my question was, do you think these models are relevant according to the fact that these rodents have immature gut at birth, and which is not the case for the infant form?
Yeah, actually, I would have a slide regarding this. It's the last slide of my presentation that I didn't show. So, but I can also answer without that if it cannot be projected. So, not only the physiology, but also the gut microbiota differs drastically between laboratory animals and humans. And we have to be cautious on generalizing the results we get on inbred mice, where basically So basically, for example, this is about diet. As I mentioned that in my studies, if everything goes right, there are basically no other variables than the experimental diet, because everything else has been standardized. And that's why it makes a lot of sense that then this experimental source of variation diet, in this case, explains a lot of the microbiota. While in humans, as I briefly mentioned, that depending whether we were born vaginally or through C-section, whether we got breastfed, how many courses of antibiotics we have gone, all this will add a background variation to our microbiome. And then when we, for example, in our case, we had intervention on resistant starts, then what we noticed was that the dark bar, which is for the microbiota composition close to 70%, that means that how much of the total variance in the microbiota was explained by the individual. So who gave the sample? And this is very high, while when we asked that how much the experimental diet explains of the microbiota variation, that is around 10%. It's a bit higher when we look for the production of short chain fatty acids. So again, the function we can affect a bit more effectively with diet. But the point I want to make, and also the answer to your question, is that laboratory animals live in a highly standardized conditions, and also something which also applies not only pharmacological trials but also dietary trials, that in many cases actually the interventions are not on physiological doses, that a high excess of the test product is given. And as I mentioned, that for example, the dominant microbiota members are completely different between mice and animals. So they are useful on identifying mechanisms, but we have to basically find a way to reproduce the same experiment in a way or another in humans.
Yeah. Thank you for that. Please, Richard.
Can I just add? I was just going to say that one of the intervention studies in laboratory mice that interested me was showing that in normal laboratory mice, as in humans, that as males age, so things tend to get worse, obviously, from a reproductive point of view. So testosterone levels go down. It varies between individuals. Spermatogenesis gets worse, etc., etc. That's probably something we just simply accept as normal. And the same thing happens in mice. But what they showed is if they altered the gut microbiome from puberty, and that's the important point to me, from puberty onwards, then the animals essentially escape these aging effects. So in a human context, if that was reproducible in some way, obviously the microbiome will be different in humans, but from a conceptual point of view, if that is translatable to humans, then that would be something we would want to know about and to be able to induce. So, but my reservation is always, in that context, considering the differences in diet and everything else, is how translatable it actually is.
Thank you. You've heard the four extremely interesting contributions this afternoon. Any other comments from the floor? Please, microphone. Thank you very much.
Nico van Belsen, IDF. Actually, a question, and that is, we referred previously already to lactase persistence, which indeed has evolved in 1/3 of the human population within 10,000 years. Now, the interesting thing is, I'm not a microbiologist, but in theory, you could also think that the microbiota could take care of that, because they can also digest lactase. So could perhaps people speculate a little bit. of why this change was made in the human genome, instead of just having an adapted microbiota?
Yeah, I mean, what I can comment is that early in life, actually, the microbiota of infant is very high on digesting lactose. So, but it declines when the solids come into the picture. And why they don't stay on the microbiota, I cannot really answer your question as such.
Okay, thank you, thank you. Are there any other points anybody like to raise or contest?
Well, perhaps on a lighter level, I think that given the fact that we, after especially some of these lectures, some people may want to rush to the hotel in the evening, I would say one very good way to exchange microbiomes is kissing, and bacteriome. And of course, I mean, this is a prelude to reproduction. In many cases, and when you perform these activities, you have low caloric intake, and at the same time you are not exposed to carcinogens. So I would suppose that this probably the suggestion of the conference is in this particular session, kiss more.
I think on that happy note, I think we've had a really interesting and dynamic afternoon. I hope you've enjoyed and been challenged by some of the contributions. Looking ahead, it is very difficult to see where, if you like, regulatory risk assessment, with all we've been discussing in some of the parallel sessions this morning about rigidity or non-flexibility in some areas to try and take out variation uncertainty. When you put it into the context of the sort of things we've been talking about this afternoon, I think each of you have said in almost the same words that this complicates risk assessment. And I think that's one of the conclusions I take away from this session. I guess that it's the same conclusion you take away. Tomorrow, you reconvene at 9 o'clock, please, in the morning, and there are 5 parallel sessions which are ranged themes coming out of today, and then we reconvene after lunch for the final 2 plenary sessions. So thank you very much for your patience, thank you very much for your engagement and contributions, and that also I extend that thank you to those who have shown patience if you've watched it on the web streaming. Thank you for that. I wish you all a very pleasant evening, and I thank the speakers lastly for their contributions. Thank you. Well done.